Now let's think about the alveoli, which is the site in which gas exchange is going to take place. Now, in general, they're dry, but they'll have a very thin layer of water that is going to be found over the epithelium. This film is needed for gas exchange, but it can be problematic based on our knowledge of water molecules. So, water molecules are composed of polar covalent bonds. We have unequal sharing between hydrogen and oxygen, which results in that dipole moment, that partial positive for hydrogen, partial negative for oxygen. And water molecules can participate in hydrogen bonding as a result of these properties. When we have water molecules interacting with one another, we can generate surface tension. And that was the example in which we had the water strider able to walk across a lake due to the hydrogen bonding and the surface tension that existed along the water. Now, there is strength in hydrogen bonds. And when we have them in appreciable amounts, they become stronger, right? When we think about the fact that they are non-covalent, so they're not as quote unquote strong as covalent, but in numbers, their strength does increase. Now, the issue with that is that if there is so much force or too much force within these bonds, that the strength of that can cause the alveoli or the alveolus singular to collapse. So the strength of hydrogen bonding can pull those water molecules together and cause the collapsing of the alveolus, which would negatively impact the ability for airflow to take place. So to counter this problem, what will be present at this location will be surfactants. And surfactants are made up of amphiphilic proteins and phospholipids, and their goal is to disrupt the hydrogen bonding. And through disrupting the hydrogen bonding, it helps to reduce the surface tension. So as the alveolus inflates, the surfactants help with resisting compression. Now, your book goes over some discussion of what would take place when surfactant is missing or lacking, which is seen in premature infants, and their deficiency in pulmonary surfactants causes them to have a really difficult time breathing, and they would, in general, need assistance or guidance in order to effectively achieve gas exchange in that scenario. So that would be a good thing to look at within your book as a way to apply the impact that surfactants have, and also to see it illustrated as a table, right? So that you're given data on it, and then from that data, you're able to draw conclusions. So do take a look at that figure within the book as it relates to surface tension. So, concept 42.6 is really all about the process of breathing and the benefit that breathing has. And so, breathing is responsible for lung ventilation. So, when we think about breathing, it's the process of inhalation and exhalation. So, we'll go through a few examples of breathing that occur in other animals. And when it comes to amphibians, they will utilize positive pressure breathing. And in positive pressure breathing, that is where air is forced down the trachea. So an amphibian will fill up their oral cavity with air. They'll close their mouth, they'll close their nostrils, they will elevate the floor of that oral cavity, and then they will push air into the lungs. So similar to like filling up a balloon, like just forcing the air into that space until it expands. Positive pressure breathing would be analogous to forcing air into a person's lung by performing mouth-to-mouth resuscitation, but positive pressure breathing isn't the way that we accomplish ventilation. Birds have a really intriguing way of breathing, and their respiratory system is highly efficient. Now, when it comes to birds, they will have air sacs. So they'll have a posterior air sac and an anterior air sac that extends between the internal organs and into the bones. And when it comes to breathing, it occurs within 2 cycles that are broken up into these first inhalation, exhalation, and second inhalation, exhalation. So in cycle 1, what we have first is air heading into the trachea, and that air is going to head towards the posterior air sacs, which will expand as they fill with air. And it is then exhaled in step 2 into the lungs. And during that time, the posterior sacs will deflate. Now, cycle 2, which will start right here, air is drawn from the lungs, and it will head towards the anterior sacs, which will expand. And then it will be exhaled from the anterior sacs into the trachea. And the movement of air is always in the same direction, so posterior to anterior, and the cycles take place simultaneously. So during inhalation, fresh air goes into the posterior air sacs at the same time that air from the previous breath that was in the lungs moves into the anterior air sacs. During exhalation, the newer air moves from the posterior air sacs to the lungs at the same time that the air in the anterior air sacs is exhaled from the body. And having this unidirectional airflow is really going to maximize the efficiency that birds have. And they're able to extract a really significant amount of oxygen from the air as compared to what we accomplish. And that's beneficial for the heights that birds can take because the amount of oxygen availability diminishes as altitudes go up. So, what's taking place during inspiration? So, during inspiration, there are muscles that are contracting. Now, the muscle contraction is important because the muscle contractions are going to change the volume of the thoracic cavity. Now, the thoracic volume, the space that's in there, will increase. The lung volume will increase because these lungs are expanding. Now, with that increase in volume, there will be a decrease in pressure. So you don't have to really worry about the term intrapulmonary. This I pulled from Anatomy and Physiology slide. Just think about pressure within the lungs goes down when volume within the lungs goes up. Now, as the lung volume increases and the pressure within the lungs goes down, it goes down to such an extent that the value of pressure seen within the lungs is lower than that of the surrounding environment. Air wants to flow down its pressure gradient, and so as a result of this change, air can enter the lungs. So that's the process of inspiration. Now, when we think about expiration, this is more of a passive process in which the muscles are going to relax. The thoracic cavity volume will decrease. You don't have to worry about the elastic recoil aspect. And as the thoracic volume decreases, the lung volume decreases. And when we see this decrease in volume, we see an increase in pressure. And with that increase in pressure, the value found within the lungs exceeds that of the surrounding environment. And so as a result, air will flow out. So we're capitalizing on changes with volume, how that impacts pressure, and how that influences the pressure gradient or the desire to go down that pressure gradient. Now, in between breaths, that will be a time in which the pressure that we find within the lungs is equal to that of the surrounding environment, and no pressure gradient exists there. And as a result, we would not expect to see any air movement. So this is an add-on from the inspiration expiration slide. because it gives us a sense of what the diaphragm is doing and what the rib cage is doing. And so what we can see is that as we inhale and the muscles are contracting, we're opening up space within the thoracic cavity. The diaphragm is contracting and it's going down. And the diaphragm is what serves to separate the thoracic cavity from the abdominal cavity. That's in this lower portion here. So as we breathe in, the diaphragm comes down, rib cage expands. We're filling up all of this with space. And in filling it up with space, we are going to decrease the pressure, and that's going to allow for air to flow in. Now for exhalation, as a reminder, we have relaxation. And so that is going to decrease the amount of space that's seen within the thoracic cavity, and the diaphragm, as it relaxes, moves upwards. And in moving upwards, we can see now that there is less room, there's less space. So diaphragm positioning contributes to the amount of volume that can be found within the thoracic cavity. And through this change, through this decrease in volume, there is an increase in pressure, which promotes the movement of air out of the body. So this is really adding on the differences in terms of diaphragm positioning. We can take a look at the rib cage. So it's really providing a more visual approach, whereas the other image was focusing more on pressure fluctuations and how that impacts this process. And we can use inspiration and expiration interchangeably with inhalation and exhalation for this class. Now, when it comes to this information, which is about pulmonary volumes and capacities, this is essentially providing a metric about pulmonary ventilation that we can use as a way to assess the severity of certain types of respiratory diseases. So we'll go through what these values mean so that we can understand any deviations from these, what that might indicate about an individual's ability to go through these events. So our first one is tidal volume. In tidal volume, we're thinking about the amount of air that is inhaled and exhaled in one cycle of normal, quiet breathing, and that's about 500 mLs. Now, if we go beyond those 500 mLs in either direction, so inspiration or expiration, that would be called the reserve volume. So we see for forced inspiration that the amount that we can forcibly inhale is more than what we can forcibly exhale. And what that essentially means is that even when we force expiration, there is still a volume of air that remains in the lungs that can never be voluntarily exhaled. That's the residual volume. In addition to these green arrows, there are some measurements that are based on capacities, and those involve formulas. So in thinking about, and by formulas, I just simply mean like adding or subtracting something. Now, one is vital capacity, and vital capacity is the maximum ability to ventilate. So that's encompassing inspiratory and expiratory and tidal volume. Okay, so this entire thing, right? And it's including the area that's in the little dark blue rectangle, and then as much as we can force out and as much as we can force in. So that's your vital capacity. So that's the summation of all of those events. And that can provide information about pulmonary health. For inspiratory capacity, you're getting information about the max amount of air that can be inhaled after a normal tidal expiration. So this value focuses on tidal volume and inspiratory reserve volume. So that's the inspiratory capacity here. So we're starting from tidal volume, and then the max that we can force in through inspiration, okay, that's going to be the inspiratory capacity. For functional residual capacity, this value represents the air in the lungs after normal quiet breathing, so in the absence of maximum effort. And that's why we're including the expiratory reserve volume within this calculation. And for total lung capacity, this value tells us the maximum amount of air that's found within the lungs. So breathing is an event that we don't need to think about, and our body is working to maintain homeostasis. So there is control mechanisms in place. So in this example, we can see that we have normal blood pH, which is about 7.4, and that's going to be our set point. Now, in this example, we fall below our set point and our blood pH goes down due to an increase in carbon dioxide. And they provide the example of exercise. So during exercise, there is an excess production of carbon dioxide, metabolic waste, because we are trying to churn out or generate as much ATP to feed those tissues. And in doing so, we generate carbon dioxide. And carbon dioxide is going to react with water and lead to an increase in the concentration of protons. So that pH shift can be detected by different things. So the medulla oblongata can detect these changes in pH. There are sensors on the carotid arteries and on the aorta that can also detect changes in blood pH. And these responses can ultimately signal to the medulla oblongata that it should regulate the rate and depth of breathing. And as a result, what we'll see is that signals from the medulla will cause the muscles to change, right? And so in the diaphragm to go through this process of contraction and relaxation in such a way that the rate and depth of ventilation goes up. So we're breathing more so that we can take in more oxygen, get out more carbon dioxide. And in doing so, the carbon dioxide levels within the blood go down and the pH will come back up to that normal value. So this is just one example of how we can control breathing within our body and how our body will respond in order to ensure that our blood pH does not veer towards too acidic or too basic.